All right. Hello, everyone, and welcome back to part three of our lectures for this week. Let's go ahead and hop into this one. So, we're going to be covering today the expanding frontiers of cognitive neuroscience. Uh, this is relevant to me. This is actually, as I was trying to decide what kind of program I wanted to go into, I was really between cognitive neuroscience or clinical neuropsychology. So, let's try to understand this a little bit more. What is cognitive neuroscience? It is the study of the neural basis of cognition. Its broad definition includes actually some of that neuropsychological testing. It compares the effects of injuries to different brain regions on particular psychological tests. And there's a lot of these new imaging techniques that are being used by cognitive neuroscientists as well to map the human brain. It also allows social psychologists and social neuroscientists to explore how the brain mediates social interactions. It can also be used to aid economists in discovering how the brain makes decisions. So, really, the field of uh, neural imaging has just exploded in recent years, and there's so much research is going on about it. I think it's an area that is extremely interesting, and there's a lot of fruitful information that can come there. What also tends to happen, though, is that people have this tendency to want to ascribe certain things to certain brain regions because it just lights up on the uh, imaging as well. So we need to be a little bit careful about that to some extent. But there's just a lot of really rich information that does come from the neural imaging, and it can be used in just so many contexts. So, like I said, one aspect of cognitive neuroscience and really the more clinical side of this is done through clinical neuropsychologists. And typically, their role is to conduct these neuropsychological assessments. Now, neuropsychological assessments, this is the systematic evaluation of cognitive functions of an individual who may be suffering from neurological or psychological impairment. It's really moved from a diagnostic role to a rehabilitation tool, especially in cases of chronic disease, and it's been influenced by these new statistical methods. Really, the way that I describe it is if you're going to see a neuropsychologist, why would you see them versus a neurologist versus getting brain imaging done, anything like that? And to be clear, you're probably not just seeing a neuropsychologist independent of those other specialists, but their role in this is with the neurologist, they could get some of the imaging done through an MRI or fMRI to get a really clear structural image of the brain. I like to say, think about that as if you're driving down the road and you see a business or a restaurant that's getting a nice kind of look at that restaurant on the outside. You say, oh, I think that looks really nice. I like the outdoor seating area. It seems like they play cool music or the lighting looks nice in there, whatever it may be. The neuropsychological assessment, that is really trying to break down to the nitty gritty of, okay, with that restaurant, how are the operations? Are people able to get their food on time? Does it taste good? Is it working well? Is this business functioning well? Is this person's brain functioning well? Now, like we know, sometimes you can drive past a restaurant in the outside, it may look really fancy, really nice. But you go there, you eat, and even though it looks like it would be great, there's some really pretty big problems there. The food doesn't taste good. It shows up on Kitchen Nightmares with or Gordon Ramsay. Um, and it's a problem. And other times, the outside of the business doesn't look that great. It looks like there's some problems, but you try the food, and you are just blown away. It's really delicious. It's going great, working well, and the person is functioning very well also. So I think that is the closest metaphor I have to talk about that. It's really trying to understand of all of the thinking skills, what are the individual's strengths and weaknesses, and what are that person's strengths and weaknesses relative to other individuals like them. Even when brain pathology is clear, there's a large variability in the cognitive effects of any type of neurological disease or condition. And that's because nobody's brains are identical, and the brain also doesn't function in a vacuum. There's all other contextual factors that take place as well, right? That need to be really considered when understanding somebody's functioning. Cognitive functions, they're not only influenced by the nature of the pathology or dysfunction, but also by things like psychological factors. So there are a lot of people who will come in for neuropsychological assessment and they're saying, I'm having problems with my memory, I'm slipping up, I'm making mistakes, I'm having all of these issues that are going on. And what can happen is maybe they have depression or anxiety. It's really, really hard to remember things if you're feeling really, really sad and down and you're hardly getting out of bed. Um, Remembering to, like, I don't know, get to an appointment, that's just not even a priority. It's not that they're not able to remember. Their brain has the functional ability to be able to do that. It's because the depression is such a barrier that it's getting in the way of them being able to actually functionally perform in that kind of task. And so then in that case, you're probably not going to treat that person for a dementia. You shouldn't. Or treat that person's memory. You're going to primarily try to treat some of their depression. There's also these socio-environmental factors. So what about living arrangements? Uh, there's times where people will go and they lived 50 years of their life in this house with their partner. Their partner passed and they moved to a retirement community or to a new house to be closer to their kids. And it's this totally new environment, totally new place, totally different from the world that they had lived in for the past 50 years. Different culture, there can be finance stressors or whatnot, just different financial positions people may have, a variety of different stressors that can be going on in life, the stability of family or work or relationships. And so when there are these neuropsychological assessments that are taking place, it's really trying to take this extremely holistic context of the person's life to understand, well, what is happening and what can best explain this. Now, one form of some of this testing is the Cambridge Neuropsychological Test Battery, or the CANTAB. It allows for tests to be administered via a touchscreen computer. It's contributed to the development of other batteries to be integrated with neuroimaging results and online mobile-supported devices. So, one major advantage of computerized batteries is that an individual's scores can be compared to a growing database of scores from thousands of people from diverse psychosocial backgrounds, such as race, ethnicity, education, gender, things like that. Although, I mean, to be fair, there's hardly any neuropsychological assessment testing that is not making those comparisons. That's really, if you're going in for good neuropsychological evaluation, your your test performance is going to be compared to other people, certainly of your age and education level, because you need to make some of those comparisons to actually understand, well, how is this person doing? Because we're probably not going to expect somebody to perform as well on a test of your vocabulary if you completed graduate school versus if you did not complete high school. And that's mostly to say that there's just a functional difference in the type of experiences that you've had, and that alone can be a pretty major factor. And so it wouldn't be fair to say that, okay, these people, we would expect them to perform the same, because if the person who didn't complete high school, if they didn't score as well in a vocabulary as somebody who didn't, or who completed a graduate degree, and we end up saying, oh, okay, they're really, really impaired in their vocabulary functioning, well, that'd be pretty inappropriate, right? Because we want to have a different level of expectation for this individual based on the education level that they had growing up in life. Meanwhile, for the person in graduate school, if they performed as well in the test as kind of the average person who did not complete high school, and we said, oh, okay, I mean, that's pretty average performance. Well, for somebody who completed a graduate degree, it's probably not. We would probably expect that they would have started at a higher level based on the level of educational attainment that they had. And so then we want to try to understand, well, why is this slower than we might expect for somebody who completed graduate school? So there's just a variety of things that can be used there. I'll step off my soapbox now, but I think it's all helpful to know that a little bit, some of that insight. As far as mapping the brain, that's a huge question. There's this Brain Connectome project, which was a task where so many researchers came together. The goal is to try to map a complete structural and functional fiber pathways of the human brain in vivo. The goal is to provide a reference atlas for those seeking to understand human brain function and dysfunction. So we have a really, really clear picture of what is uh, that like the pathway and the structure of the typical human brain, and then what are differences then from that. So we can have this like baseline to compare everybody else to. This is really similar in some ways to the Human Connectome project, which combined diffusion tensor imaging and functional connectivity, MRI, to plot these connections in the living human brain. And other studies outside of the HCP seek to describe specific processes and disorders using mapping technology. So in these figures here, in figure A, we have the Human Connectome Project's multimodal parcellation map based on the FACT technique. This technique identified 180 distinct regions. And in figure B, we have an estimate, of 17 cortical networks based on the fcMRI data from 1,000 patients. In this picture, each color represents a network. Now, some networks, such as the blue auditory areas in the temporal lobe, are localized, whereas others are widely distributed, such as those yellow regions, which reveal the prefrontal posterior parietal connectivity. When using the fMRI in brain mapping, it's really using this resting state fMRI to measure functional correlations between brain regions. It identifies these consistent patterns of connectivity or nerve tracks in the brain. It does not measure the static anatomical connectivity, but instead uses temporal or like a time-based correlations between neurophysiological activity in different regions to infer functional connectivity. So some of the networks of the social brain, there are these four proposed social brain networks. The red areas represent the amygdala network. The light blue is the mentalizing network. Dark blue is the empathy network, and green is the mirror simulation action perception network. There's also the default network. So brain network, that's the brain network involving regions of the frontal and parietal lobes. Higher resolution imaging demonstrated the default network is not a single network, but it's composed of at least two parallel networks, including adjacent dorsal and ventral components. Sangyal Lee and colleagues showed that components can be functionally dissociated, and the ventral region responded to the vividness of the imagined events, whereas the dorsal region responded to their valence. Valence, we're talking about kind of like the affective quality. Tractography using diffusion tensor imaging. So tractography measures actual neuroanatomical pathways that can be related to specific traits. This can be done quickly on many living brains, and the measurements can be made simultaneously in the entire brain. It complements the networks mapped by the fcMRI, and it measures actual neuroanatomical pathways that can be related to specific traits. There's also this hyperconnectivity, which is the increased local connections between two related brain regions. Now, what we have in this figure here uh, is we have this color overlaid on the diffusion tensor images, which demonstrates the tracks between the superior temporal gyrus and the middle temporal gyrus of three individuals. Participants A and B had more accurate and less accurate perfect pitch, respectively. Case C did not have perfect pitch. The tracks that connect the regions of the left hemisphere in orange are larger in participant A and B than in C. The tracks colored purple connect the areas in the right hemisphere. Now, another thing to be just kind of thinking about when we're talking about cognition in the cerebellum, uh, the cerebellum accounts for 80% of the brain's neurons, which, to be honest, when I had learned that, that fact really blew me away. Um, the fact that 80% of the brain's neurons are in the cerebellum. This is the extensive neocortex. The cerebellum interconnections include the prefrontal cortex, Broca's area, neocortical regions with sensory or perceptual functions. The cerebellum is critical in producing fine movements and perception, and it's associated with working memory, attention, language, music, decision-making processes, and more. RS-fMRI has identified cerebellar networks that are related to similar cortical networks. It's not yet clear how these cerebellar and cortical networks interact. Still, in the coming decade, will undoubtedly deliver an abundance of new information on this topic, likely allowing researchers to develop a model of cortical cerebellar functions in cognition. In some of the parcellation of the cerebellar resting state networks, the networks are identified with the same colors as those for the cerebral cortex, and that were is in figure 1512b. It's unclear how these networks interact again, but I think in the coming years there's going to be a lot of work on this. In one of the recent conferences I had gone to, there was actually a talk that was discussing cognition in the cerebellum, and I really think that's going to be a hot area of research in the coming years, trying to understand what is the cerebellum's role in all of the rest of it. So I think we touched on this briefly, but we'll talk about it a bit more. Social neuroscience. This is an interdisciplinary field that seeks to understand how the brain mediates social interactions. It combines cognitive neuroscience tools, especially some of that neuroimaging, with abstract constructs from social neuroscience. Now, one of those concepts that's particularly interesting is an understanding of oneself. Humans and other animals can recognize themselves in a mirror. The brain activity increases in the right lateral prefrontal cortex and the lateral parietal cortex with recognition of our own versus the face of a familiar other. When participants are asked to determine whether trait words or sentences are self-descriptive, brain activity increases in the medial prefrontal regions. Now, when we're trying to understand others, there's this other really major concept called the theory of mind. Inferring minds of other animals in part by observing their behaviors, and in the case of people, by listening to their words. Understanding that others may have different feelings and beliefs from our own. So, really, the way that I think about the theory of mind is it's what I think you're thinking about. And even if it's what I think you're thinking about me, for example, this kind of conversation comes up a lot. I think if you're having an argument with a partner, for example, and you say something like, you just think I'm lazy or something like that. Well, if that comes up, you just think I'm lazy. So I, Bradley, think that you, person B, thinks that I, Bradley, am lazy. So it's this me thinking that you think about me, um, in this really interesting way. And that's a it's a heady concept. It's one that's difficult to kind of wrap your mind around, but it's worth thinking about. And I think especially in thinking about things like compassion, empathy, and whatnot as well. It's really this capacity to understand others that can be inferred from the presence of empathy. Empathy is correlated with this increased activity in the medial prefrontal cortex. Concept of self-regulation. This is the ability to control our emotions and impulses as a means of achieving long-term goals. It's the capacity to put feelings into words, and there's the expectations of how a stimulus might feel. Prefrontal regions are critical for self-regulation, and children are often poor at self-regulation, which probably reflects the slow development of prefrontal regions responsible for impulse control. So, our understanding of ourself and our social interactions link together as a single mental action. When we express attitudes, including our prejudices towards ideas or human groups, brain imaging shows activation in prefrontal, anterior cingulate, and temporal parietal junction regions. Conclusions drawn from these studies tend to be controversial because the ability to replicate findings is central to the scientific method, but replication is rarely done in fMRI studies. And part of that's just because of how hard it is to do a lot of these fMRI studies. So a lot of people don't want to just repeat what somebody else had done. They want to try to do something new. The social cognitions are associated with activation in specific brain regions, especially the prefrontal regions. The prefrontal activity produces our social cognitions, just as activity in our visual regions produces our visual perceptions. So the medial prefrontal cortex uh, and the temporal parietal junction activity then produces our social cognition, and the medial prefrontal cortex and the temporal parietal junction correlated activity and activity in other regions in the default network commit new social information to memory during rest. Then I will just do one slide talking briefly about neuroeconomics. This is an interdisciplinary field that seeks to understand how the brain makes decisions, finds ideas from economics, psychology, and neuroscience in an attempt to explain decision-making processes by studying patterns of brain activity as people make decisions. Two neural decision pathways influence our choices. There's the reflective system, which is a deliberate, slow, rule-driven, and emotionally neutral um, kind of system. And then there's the reflexive system, which is fast, automatic, and emotionally biased. Epigenetic factors probably contribute to the developing balance between these pathways. And there's this general assumption among neuroeconomics that two neural decision pathways really influence our choices. And there's, like I said, the system one and system two that take place. I think probably a book, if you're wanting to read a dense read, it's called Thinking Fast and Slow by Daniel Kahneman. It's a very, very long book, very dense, but very rich. It talks about these systems one and two, and it also talks about all the problems that can take place and do take place in all of us in that system, how it can be hacked and um, used to kind of mess us up in our thinking. So if you want to learn more, I would point you in that direction. That is the end of part 3 of our lectures for this week. Thanks so much for sticking around, and I will see you around next time.